Hello, everyone. My name is Nate. I own the website MCAT, where I have a whole bunch of free resources such as cheat sheets, graphics, notes, tips, an equation sheet, Anki decks, and links to other great resources as well, the whole nine yards. I also offer tutoring on my website as well. But today I woke up a little bit grouchy, and so I decided that I'm going to make a passage skills video, and I'm going to let myself yell at you a little bit and give you some tough love. There's a lot of really super common mistakes that a lot of my students have made throughout my few years of tutoring for the MCAT. So I decided to make this video to hopefully help you start recognizing and avoiding making these mistakes a little bit earlier in your studying progress. Today, specifically, we're going to be working through a cancer pathway passage, getting some good practice with all of those really complex, long, and annoying pathways that you might see on the MCAT. So just to start off, I'm going to give you a pretty long list of my general tips for going through MCAT passages, specifically within bio and biochemistry. Typically, all of my tips you can kind of extrapolate to the other sciences as well. Cars is slightly different and kind of its own beast. But for the sciences, especially bio and biochem, as far as timing is concerned, I usually tell students that a good rule of thumb to kind of stick by is adding the number 3 to the number of questions per passage. Each passage will tell you how many questions it has at the top. So if it's a 4-question passage, that means you should probably be spending somewhere around 7 minutes on it. That's a pretty easy way to keep track of time whenever you're going through the bio and biochem passages. And that's usually just my initial recommendation. Whenever you're actually reading a passage, especially when you get to the actual experiments, I always ask myself, why are they mentioning this? What was the point of them bringing this up? Are they trying to continue on with the other content that they've introduced? Are they trying to prove a point? Are they trying to disprove a hypothesis? What is the point of mentioning this specific thing, this specific sentence? I also like to identify keywords that you're already supposed to know. Going into this exam, there's quite a bit of content knowledge that is pretty well established that you need to know. For example, you need to know what a tumor suppressor gene is. So if they mention a tumor suppressor gene, that means they're probably going to be asking you about them. So then reading through the rest of the passage, kind of with the lens of, okay, this is kind of what this passage is going to be asking about, should kind of make it easier for you to locate in your brain all of the information that is going to be relevant for the questions that might be showing up. Additionally, this is pretty common advice, but you're looking for the Spark Notes version of the passage. This is not a dissertation. You're not trying to memorize every single word. Pretty much all you're doing is, if they're defining new things, just make sure you have a super basic understanding of whatever they're defining. And if it's an experiment, make sure you understand the basic results of the experiment as well as why they conducted the experiment. Everyone's going to be reading at a different speed, and that's perfectly okay. There's not a single speed that is the best speed that I recommend for everyone to be doing, because everyone's going to be reading at a different pace. But in general, you want to try to be pretty much done with going through the passage within about 3 minutes at the max. This should give you around a minute to a minute and 15 seconds per question, which should be plenty. As far as highlighting is concerned, I don't really have super specific advice. I'll give you examples of what I did and why I like to highlight certain things. But quite honestly, you should just highlight whatever works best for you. And most importantly, if you start noticing that there's certain phrases or certain words that you keep missing whenever you're going through passages, maybe you then make it a point to start highlighting those words in the future. For example, in Cars, I used to always miss all of the tone reversals, like whenever the passage would say however or alternatively, something like that. I used to miss that, so I would make it a habit to start highlighting that so I wouldn't miss it in the future. So just the things that I would highlight any of those words that I knew were supposed to be part of my content knowledge. Like I mentioned earlier, this is just going to kind of prep you and put your brain in the right mindset for whatever that passage is asking about, so that if the first paragraph brings up antibodies, you know you're working with the immune system and not electrolytic cells or something random like that. It's also a good idea to highlight any relationships. So if something inhibits something or promotes something else, always a pretty good idea to highlight those. I like to highlight anytime they explicitly mention results, whether they're expected or unexpected. And then whenever you're going through graphs, this is not necessarily something that you can highlight. You don't want to understand every bit of information that the graph is telling you, but I usually look at the general trends or outliers of the graph. Maybe there's one line that is going a completely different direction than the others. Then just kind of asking myself really quickly, maybe why do I think that occurred? I'm also really quickly going to edit this line because I want to make sure that you understand that you have to look at the axes. The axes are often where you're going to be finding a lot of information that can help you clarify what you're looking at on the graph, or maybe they did something weird where they inverted the y-axis or something like that. It's a really good idea to make it a habit of looking at the axes of the graph. Sometimes the description of the figure can help you out as well. So it's just a good idea to make sure you're at least glossing over all of the information that they give you and you're not missing out on something important. Sometimes they'll give you specific info about the lab techniques or experimental procedures that they use, like maybe mentioning something about how the study was a double blind or something like that. There's probably a reason that the makers of the MCAT included that sentence. Maybe they're going to ask you about what a double blind is later in the question, something like that. Or maybe they'll mention that they did SDS page or something along those lines. So highlighting stuff like that just makes it harder for you to miss that information. And then finally, anything else you deem important within your own brain is something you should feel free to highlight. You want to use highlighting much more sparingly rather than liberally. You want to use it to help you identify the key things rather than just highlighting the entire paper and then having just a giant yellow blob all over the left side of your screen. So you obviously might have your own strategies and things that you highlight, whether it's from your own knowledge or your own course or something like that. Obviously, like I mentioned, just do whatever works best for you. But the most important thing is just to make sure you're not spending too much time highlighting and using too much of it, because then it kind of loses the entire point. As far as actually how you should be approaching the passage, the first thing that I'm going to say is that you should be finding a strategy that works best for you. There's several different really common strategies that people use, but there's never going to be a one-size-fits-all strategy. I highly recommend trying out different strategies, especially at the start of your MCAT studying, because you might be surprised. Maybe there's a strategy that you don't think will work that ends up working for you. For example, some people really like to just write a quick little summary of each paragraph whenever going through them. If you are going to do this, I recommend trying to make the paragraph less than a sentence. That way you're not spending a lot of time writing. But sometimes this just helps people organize the information, and it forces you to make sure that you're actually understanding the information. This was something that I did that kind of surprised me because I wasn't expecting it to help, but actually reading through the questions first was a huge benefit for me. It's one of those things that not a lot of people like to do, but for some reason it just clicked for me and it made it easier for me to understand what I needed to be looking for in the passage. That's the idea of reading the questions first. If you are going to do this, you need to make sure that you're doing it pretty quickly, but like I mentioned, it may help kind of guide what you should be looking for in the passage. Some of my students have liked to skim the passage really quickly and then read it again, basically just giving yourself 2 complete exposures to the passage right off the bat. Maybe that's something that you find you like. And then some of my students only skimmed, especially for Chem and Phys. It might sound crazy, but some students literally don't even really read the Chem and Phys passages. That doesn't work for a lot of people, but it works for some people. So the important thing that I'm going to mention is that you should find a strategy that works for you. It always kind of frustrates me whenever I see people trying to say that you should always be reading the question first or something like that. Not every strategy is going to work for everyone, so just try on different pairs of pants until you find the ones that fit. You also, of course, are going to be given a little booklet that you can be taking some notes in. Obviously, there's rules that go into how you can use the notebook, like you can't erase anything, and you're given a Sharpie to actually write in it. But you've got plenty of space to do whatever you want in there. You're never going to run out of space, and even if you do, you can ask the administrators of the exam if they can look over it and then give you more space. But basically, one of the common questions that I get is, when should I start using my notebook, my noteboard, or whatever you want to call it, whenever I'm actually doing the sciences? Obviously, you're going to need it to do math and things like that. But whenever you're going through the passage, especially in bio and biochem passages, I recommend writing things down whenever your brain can no longer keep track of things on its own. That probably sounds like a pretty obvious and stupid thing for me to say. But basically what I'm trying to say is that if you get like a pathway, like we're going to be looking at today, you don't want to be trying to understand the entire pathway in your brain. Once they first mention that something promotes something else, I highly recommend writing that down immediately. So the common places that I noticed that it really helped me to start writing things down are those passages where they give you a whole bunch of new terms that you're not really used to, or you do have one of those really complex pathways where there's a lot of things promoting or inhibiting other things. So those are just my very broad general tips for going through bio and biochem passages. Whenever we actually go through this passage here in a second, you're going to see my much more specific advice, especially in regards to how to actually answer questions. But maybe in the future, I'll give you an example of how to go through each one of these strategies. Who knows? If you have a specific strategy or type of passage or something that you want me to make a video about how to approach, you can always just suggest things either in the comments or there's a place that you can contact me on my website. All right, with that all being said, I just want you to keep in mind that I'm going to be going through this passage as quickly as I can with the intent of actually showing you what I'm thinking in the exam, not necessarily trying to explain everything that's going on. I'm going to be just as confused as you are in some parts, but that's okay. It's okay to be confused about something. Whenever you're going through the passage, you're not going to understand everything. So just as long as you get the main idea, you understand the logical path of the pathway, you're going to be fine. You'll see what I mean. So the passage starts off with talking about this mammalian circadian rhythm. It's a tightly regulated endogenous cycle, that's one of those words that you can highlight if you want, that governs physiological and behavioral processes over a 24-hour period. I already know about the circadian rhythm, so I'm not too worried about that. This is just fluff. It's just introducing what we're talking about in the passage. At the molecular level, this rhythm is maintained by a transcription translation feedback loop involving a set of core clock genes. BMAL1 and PER2 are these genes that they're going to be talking about in this passage. Okay, so here's my context for what we're talking about this passage. They're talking about genes, transcription, translation, central dogma. It's going to be a pathway passage. So now I know that that's kind of what we're doing when we're going through all this info. In the morning, humans express cortisol, part of this cortisol awakening response. I'm going to highlight cortisol because that's a hormone that you need to know about, which is partially coordinated by one of those proteins they talked about earlier, BMAL1. So because I know that this is going to be one of those complex pathways, I'm going to go and start writing things down. And I'm just going to write that BMAL1 promotes cortisol. Maybe it's not a direct promotion, but this sentence just told me that whenever BMAL1 is there, that's going to help us actually release cortisol. Throughout most of the body, BMAL1 expression alone promotes genome stability and proofreading, meaning that it's one of those genes that kind of stops the cell cycle to check on it to make sure everything's going okay. We call these tumor suppressor genes. So that's one of those things that I'm going to write down. BMAL alone is slowing down growth, throws up the stop sign to make sure everything's all good. Again, we call these tumor suppressors. I think BMAL1 is a tumor suppressor, but maybe I'm wrong. We'll just keep reading and see. Until it is bound to a circadian protein called clock. The formation of this BMAL1 clock heterodimer, maybe they're going to ask me about what a heterodimer is, causes the promotion of growth factors and mitotic genes such as ABK, an enzyme responsible for ensuring chromosome alignment in metaphase. So BMAL1 alone seems like it's probably one of those tumor suppressors. Once clock binds to it, it basically turns off that tumor suppressor and allows the cell to grow. Seems like that's what's going on. And then the signal that they're going to give us that the cell is growing is ABK. One thing that you'll notice too is that I typically highlight the acronyms instead of the full words. Just the reason for that is typically whenever they ask about things like Aurora B kinase, in the questions, they're just going to say ABK. They're not going to say Aurora B kinase. Obviously, either one's fine. I just think it makes it a little bit easier for me to find. So that's the second paragraph. This is the first paragraph that we're actually getting some substance that they're probably going to be testing us on. There's not really any info in that first paragraph that we can really craft into some sort of question. I'm also just going to write down that last little connection we made there at the end, that the formation of that BMAL clock heterodimer promotes cellular growth as well as that ABK protein. As the BMAL1 clock heterodimer accumulates throughout the daytime, as its concentration grows, that's going to promote the expression of circadian gene PER2. So now I have another relationship I can write down. High amounts of the heterodimer promotes this new protein PER2. Following translation, you could highlight translation if you wanted to, but I already know that this entire passage is talking about translation. That's not something that I'm going to be missing because we're talking about the whole central dogma here. Following translation, PER2 in the cytosol will associate with cry proteins. Who knows what that is? That's another one of those things that you could highlight because it's something new that they're introducing. This complex is going to translocate into the nucleus where the complex will inhibit the expression of BMAL1. So the BMAL1 heterodimer with clock is going to promote PER2. And once PER2 is promoted and associates with its protein that it's going to associate with, it's going to go back and inhibit BMAL1. This is one of those classic negative feedback loops. One gene promotes the production of the second gene goes back and inhibits the formation of the first gene. So just going to highlight that really quickly. Going to keep reading for a second and then we'll go back to writing things down. Inhibition of BMAL1 in cells aids the SCN, whatever this thing is, stimulated release of melatonin from the pineal gland, a hormone that promotes sleep. You do need to know kind of what melatonin is, what type of hormone it is, what releases it, as well as what it does. That's one of those things that you do need to know. But apparently this thing, the SCN, the suprachiasmatic nucleus, is helping us release melatonin. And the way that it does that is through this inhibition of BMAL1 that PER2 is doing. So as PER2 levels accumulate, that's going to promote the inhibition of BMAL, which is going to allow us to promote the production of melatonin. Beyond their roles in circadian timing, accumulating evidence suggests that BMAL1 and PER2 exert significant influence over cell cycle progression, DNA repair, and apoptosis. Okay, probably about to start talking about cancer. To study the roles of circadian genes in tumorigenesis, there it is, tumorigenesis just means the genesis of a tumor. Researchers collected tissue samples from the colon of patients with colorectal cancer. They're calling this group B, and patients lacking colorectal cancer, you're going to call that group A, and measured the expression levels of both of these proteins they've talked about earlier. And the results are shown in figure 1. So anytime you're going through an experiment, it's a good idea to identify which group is the control, because you know what's supposed to happen with the control. And then once you've identified which group is the control group, you're comparing the other groups to that control group. So group A is going to be the control group. They're the ones lacking colorectal cancer. So we're going to be comparing group B to group A. One other thing that you could also highlight from this sentence is this idea that they're collecting tissue samples. You might be asking, like, why is that important? The reason that that's important is because that's going to help me easily identify that this is an observational study. They're basically just grabbing tissue samples from people and just measuring how much of these proteins they have. One of the reasons that I like to identify what type of study they're running is because the type of study can help us understand whether or not it's a correlational or a causational study. I could dive into all of that right now, but just as a spoiler, there's going to be a question that's going to bring that up. So now I'm just going to show you really quickly how I go through the figure. You might have noticed that I just snuck in a slight change on the figure. Just pretend like you didn't notice that. Where on the y-axis, it says normalized minimum, if I could read, normalized maximum 24-hour concentration. It's like, what in the hell is that? So if it's normalized, they're basically just stating that we're basically adjusting the concentration levels to where a normal level is considered 1. So maybe a normal level is 100 milligrams per deciliter or something like that of PER2. And so if you have that, then on a normalized concentration scale, we would just say that your concentration is 1. Your normalized concentration is 1. So the maximum concentration of PER2 in the control group, which is group A, is right around 1. Same thing for BMAL1. They're both right around 1 for their maximum concentration throughout the course of a day, which that makes sense. We would expect them to be normal. They're the control group. But notice in group B, both of them have less expression. So that's what figure 1 is telling me, is that if you have cancer, which we know that the people in group B do, that having cancer is correlated with a decrease in PER2 and BMAL. The reason I say correlated is because we can't prove necessarily that colorectal cancer is causing a decrease in both PER2 and BMAL. We just know that for some reason, people with cancer end up with less PER2 and BMAL. Maybe there's something in the middle that's actually causing it or something like that. Basically, what I'm trying to state is that this is a correlation, not a causation. On the MCAT, observational studies are pretty much always going to be correlational. So that's one of those things that often confuses a lot of people whenever we get to questions. You'll see what I mean a little bit later. So now I'm just going to move on. I've kind of identified the main takeaway from figure 1. Maybe there's other information that I could take, like also like notice that PER2 levels in both are a little bit lower than BMAL1. That's whatever. I don't really care. Basically, I've got the main takeaway. I've compared my groups to each other. I've identified any outliers. I understand the axes. I'm going to move on with my life. In a subsequent experiment, researchers conducted targeted knockouts in 4 groups of mice, 3 of which containing at least 1 knockout of PER2 or BMAL1. So it's like, okay, well, what in the world does that mean? It's okay if you don't understand what that means, because maybe if you read on a little bit, you'll understand the experimental design a little bit more. So let's just pretend like you don't really fully understand what's going on there. The rates, I think that's supposed to be rats. That's definitely supposed to be rats. I'll probably fix that here in a second. The rats were exposed to a known carcinogen over the course of 6 months. Maybe that number is going to be important later. At which point, the percentage of mice containing a tumor were measured between all 4 groups. So they're just basically trying to figure out how much cancer did each of these groups have. So mice that contained both PER2 and BMAL1 had a cancer prevalence of 8%. If you knocked out, that's what that symbol means. That little delta symbol means a knockout. If you knock out BMAL1, that led to a cancer prevalence of 23%. If you knock out PER2, that leads to a cancer prevalence of 25%. And then if you knock out both, that leads to a cancer prevalence of 71%. So this second experiment is basically just telling me that if you knock out both, that leads to cancer. That's basically kind of proving that they are tumor suppressors. If you remove a tumor suppressor, we expect cancer to increase. So notice for both of them, once we removed them, each one separately, they individually led to an increase in cancer rate. And then together, we had a dramatic increase in cancer rate, which makes sense. That's what we'd expect. So kind of a lot going on, but it's really not too bad. I think the main thing that I want to kind of point out at this point is that you want to kind of be reading through it through the lens of a test maker, kind of thinking about what are they potentially going to be asking me about. You don't want to go down every single pathway that they could be asking you about, because obviously they mentioned heterodimers. They can ask you a whole bunch of questions about SDS page and how proteins work and stuff like that. They talked about 2 different hormones. So obviously you don't want to spend too much time on it, but basically the stuff that you should be keying into are the things that you think they might be asking about. They're probably not going to ask you about what the definition of mammalian circadian rhythm is, but they probably will ask you about what is the role of BMAL1 in cortisol responses, or what is the role of BMAL1 in metaphase, something like that. All the stuff that they're kind of bringing up that we are affecting with these genes, that's the stuff that they're probably going to be asking about. So to quickly go through my tips for actually answering the questions, especially within the sciences. Just to start, again, to avoid making a stupid mistake, you want to highlight whenever the question says not or except. All of those things with those like crazy double negatives that they do just to be annoying little, pardon my French, bitches. They're just doing their job. Quite frankly, you can't really be mad at them for just trying to do their job. But it's really annoying whenever they do that. So in order to avoid missing those types of questions, I recommend highlighting those. If you have never missed one of those in your life, fine, do whatever you want. But if you're a normal human being, you're probably going to forget about that at one point in your life. You're going to go through an entire question and accidentally pick something that's true when you're supposed to be looking for something that's false. So I recommend highlighting that. Additionally, a lot of the questions are going to have a lot of fluff. There are going to be some examples of this here in a second. But the main thing that I want you to do is just put the question in your own words. Just identify the root of the question. What is the question really asking you? So every single time I go through a series of answer choices, I approach them the exact same way. And of course, that's going to be process of elimination. It's easier to prove things incorrect than it is to prove things correct. The way that I usually word this is it's easy to be a hater. It's super easy to hate a lot of things. And so your job on the MCAT is to be a hater. You're trying to identify why you hate each of these answer choices. So on your first pass, you want to eliminate answer choices that are scientifically incorrect. This can probably get you to an answer on probably 30 to 50% of your questions, even if they're passage-based. If there is any ounce of an answer choice that is scientifically incorrect, immediately rule it out. I'm not even kidding. Don't even consider it unless it's obviously like one of these not or accept questions, but we're no longer talking about any of those. For all of the normal questions, if there's any part of any question that is even remotely incorrect, immediately rule it out, throw it out, dump it down the drain, turn on the disposal, don't even think about it. It's wrong. Scientifically incorrect answer choices will never be the correct answer choice. So once you've done that, now you're actually going to be trying to answer the question. On your second pass, I highly recommend trying to then eliminate answer choices that don't really answer the question. Your job is not only to pick a scientifically correct answer choice, but also pick one that's actually answering the question at hand. On your third pass, if there's still answer choices left, multiple answer choices left, you want to eliminate the answer choice that doesn't apply to the passage at all. So this passage that we have here is talking a little bit about hormones, cancer, the central dogma, stuff like that. So if there's an answer choice that randomly brings up sliding filament theory, there's not any logical connection to sliding filament theory in the passage, at least not one that's very clear at all. So if there's one that has a much more direct tie-in with the passage, that's probably going to be your better answer choice. Finally, if there's a tiebreaker, meaning you have deemed these answer choices to be equal on all 3 of your first few passes, you're basically just going to be picking the answer choice that's more specific. I want to note that you should never rule out an answer choice just on the basis that it's vague. You should only be ruling it out because it's vague if it's equal on every other more important quality of an answer choice. So here comes my most important advice. Your job on the MCAT is to pick the best answer choice, not a correct answer choice. This also applies to cars a little bit. Once you find an answer choice that is correct, you're going to get into a lot of trouble if you just pick that answer choice and move on with your life. Just because it's correct does not mean it's the best answer choice. And your job on this really annoying exam is to pick the best answer. The easiest way to do that is to figure out which answer choice is worse than the others. That's why you're doing process of elimination. My last bit of advice, this is really important for the timing, is that if you spent more than 2 minutes on a question, at that point, just pick your best option, flag it, and move on with your life. If you haven't gotten it after 2 minutes, it's not like you're going to have an aha moment in that second and then find the right answer. The quality of your answer choice isn't really going to change after this. This is an exam that's timed. It's also incredibly difficult, and you're going to miss questions. You do not get bonus points for getting a really hard question correct. So it's best not to waste your time on a question that you think you're probably going to get wrong in the first place. Obviously, you flag it so that it's easy to come back and answer if you do have time at the end of the exam. But again, you don't want to get caught up on the really difficult questions, which is going to give you less time for doing maybe some quite a bit easier questions later on in the exam. Obviously, you might have accommodations, so your timing is a little bit different, but I'm hoping you guys understand that you can just adjust all of the numbers and stuff on my slides just a little bit for if you have timing. So now to go back to the actual questions. The first question that we're going to talk about is asking which of the following identifies the most likely manner in which the researchers studied BMAL1 and PER2 expression rates in the first experiment. A lot of words, great, whatever. So 2 key things. Remember, I said that your job is to pick the best answer choice, not a correct one. Notice that in this question, they said most likely. So it's possible that there's an answer choice that likely provides an answer, but it's not the most likely. Your job is to find the most likely answer choice. Pick the best answer choice, not just any correct answer choice. So to start with the lessons that I want you to be taking away from all of these questions, basically we're going to be going through all of the steps that I mentioned on the last slide. The first thing that we're going to do is identify the root of the question. Basically, put the question in your own words is what I'm trying to say there. So this question is literally just asking us, how can we measure our mRNA or protein amounts? Because we're trying to measure expression rates. Expression is talking about how much mRNA are we turning into protein. That's what gene expression is. So this question is basically just asking, how can we measure that? What lab technique can we use? So whenever I'm going through these answer choices, I'm just going to ask myself that question. Will this either measure mRNA or protein? And is it going to measure an amount? So now whenever we go through these answer choices, we're going to be first eliminating any that are scientifically incorrect. We're going to be eliminating any that we know for a fact do not measure mRNA or protein amounts. So starting with answer choice A, utilizing northern blotting to identify cytosolic protein concentrations. We can immediately eliminate this. Northern blotting identifies RNA, not proteins. Northern blotting is supposed to be measuring RNA when answer choice A is saying that it's measuring protein concentrations. That is scientifically incorrect. Immediately rule it out. Just on a scientific basis, answer choices B through D are all correct. Whatever lab technique they mentioned actually does what the rest of the answer choice states that it does. So from here, we're gonna have to eliminate the answer choices that don't actually answer the question. Again, we're looking for something that measures mRNA or protein. Either one's fine. And we're looking for something that measures an amount. So then on my next pass, I can first eliminate B. We can eliminate B because SDS page is not really going to help us figure out how much of something we have. Sure, maybe you can argue that you can figure out the relative amount of something in SDS page because of the thickness of the bands. Maybe that will let you figure out one band is thicker than the other, so we have more of that band. But that's arguable. It's also not a very accurate way to measure the amount of something. And additionally, comparing answer choice B to answer choice C, Western blotting is way better of a measure of the amount of something than answer choice B. The goal of B is to just help us identify the weight of a protein, which can help us identify the protein in general. SDS page doesn't really help us identify how much of something there is. So I'm going to eliminate B, even though, again, you could argue that B might work. We're looking for the best answer choice, and right off the bat, C is a better answer choice than B. So then between C and D, we're going to end up eliminating answer choice D because identifying DNA is not going to help us identify gene expression. For gene expression, we need to know how much mRNA is being made and how much protein is being made from that mRNA. So either one of those two is fine. Just identifying how much DNA we have is not going to help at all with identifying how much gene expression we have. Our DNA is permanent. It's always there. So Southern blot is great that it will help us identify that the PER2 gene exists, but that's all it's going to do. And again, I know what your little brain is sitting there thinking, oh, well, maybe there's methylation or something like that that promotes promotion of this, whatever thing, whatever, and whatever. Again, that's arguable. We are looking for the best answer choice, and there is nothing about C that is arguable. You can't be a hater of C. There's nothing wrong with answer choice C, so that's the one that we're going to pick. All right, question 2. Which of the following accurately identifies properties of the hormones correlated with BMAL1 and PER2 respectively? So just like I mentioned, the first thing that my brain would be thinking of was basically, what is this question asking? What is the root of the question? This is literally just saying what type of hormones are cortisol and melatonin, and what are the properties of those steroid and peptide hormones. So now on our first pass, whenever we're going through trying to figure out if there's anything scientifically incorrect or not, we basically just need to make sure that cortisol is accurately being identified as a steroid hormone and melatonin is accurately being identified as a peptide hormone. Notice in the question it says respectively. That basically just means that the first part of the question is going to be talking about the hormone with BMAL1, and the second part of the answer choice is going to be talking about the hormone with PER2. It's just in order, basically. So now to actually start eliminating answer choices on their scientific correctness. Again, cortisol is a steroid hormone, melatonin is a peptide hormone. There's characteristics of each. I'm going to put those up on the screen, but I'm not going to fully talk through them. I mean, I will as I'm going through the answer choices, but I'm not going to fully define peptide and steroid hormones right now. I will say, however, just to help with the naming, because a lot of students really struggle to kind of classify the hormones at first, you can easily identify what type of a hormone it is just based on the suffix. So if it's a steroid hormone, it's going to end in izol, ogen, or erone. So like testosterone, cortisol, estrogen. Those are examples of kind of the suffixes that we see with steroid hormones. So if you see that suffix, it's just really easy to identify as that a steroid hormone. Anything that just ends with IN or has an acronym as part of its name, for example, like ACTH, those are all peptide hormones. So we can actually start eliminating answer choices because we'll just start off with looking at BMAL1, the first part of the answer choice, which is supposed to correlate with cortisol, a steroid hormone. Steroid hormones are not made of amino acids, so I can immediately get rid of that one. Didn't even look at the rest of the answer choice. First part is false, getting rid of the rest of it. B is fine because it does accurately talk about cortisol, a steroid hormone in the first part of the answer choice, and melatonin, a peptide hormone in the second part of the answer choice. C we can get rid of because it flips the two. Again, cortisol, which is the first part of the answer choice, is not soluble in the blood. It is non-polar. It's a steroid hormone. So we can immediately get rid of that. And then D, again, flips the two. It gets them opposite of what we'd expect. So this is a really easy question. It's B. Don't overcomplicate it. This is literally just asking what are the properties of steroid hormones and what are the properties of peptide hormones. That's it. So please just stop overcomplicating things. I promise if you just take like a 3% of a chill pill, you're going to be fine. All right, now moving on to the third question, we get a Roman numeral question. And I do have a specific strategy for Roman numeral questions. That might make your life a little bit easier. Which of the following statements are true considering the results of the experiments described in the passage? So just a very standard, basically, what can we learn from the passage? So first thing I'm going to do really quickly is just identify what the question is asking. Again, this is a straightforward one. It's just like, what is the passage telling us? So for Roman numeral questions, I have this strategy that I use pretty consistently. I don't really know what to call it, so I'm just going to call it the 50-50 strategy. Basically, what you're doing is you're going to look at the answer choices first. And looking at the answer choices, you'll notice that Roman numeral 3 shows up in 2 of the answer choices. So basically, if we can start with 3 and eliminate 3, we say 3 is false, then all we have to do is figure out whether or not 2 is true or false. We don't even have to look at 1 at that point. Because basically, we can eliminate B and D, leaving us with A and C both have Roman numeral 1 in it. So basically, just by doing kind of like this 50-50 strategy, identifying a Roman numeral that shows up in 2 answer choices and then either eliminating that or proving that to be correct, you're making your life a little bit easier because you probably don't have to go through one of the Roman numerals. So now we get to start going through the Roman numerals. Of course, we're going to be eliminating anything that's scientifically correct, doesn't apply to the passage or the question or anything like that. But we are going to go ahead and start with Roman numeral 3, which states that the second experiment demonstrates that the removal of PER2 and BMAL1 causes cancer in mice. So the first thing that I want to note is that you need to be really careful with correlation versus causation. This is one of those things that really confuses most students on the MCAT. We kind of alluded to this earlier, but a correlation just tells us that we know that there's some sort of relationship, but we don't know why it happens. We don't know what causes what, or even if there's a causality at all. For example, you might have seen those graphs that show that, like, for example, I think there was one that shows that like the number of people dying to being stuck underneath their bed sheets perfectly matches the number of redheads in US Congress or something. I'm pulled that straight out of my ass, but I hope you get what I'm trying to say. There's a mathematical relationship between those two things. They're correlated in that when one goes up, the other one just happened to go up, but there's no causation. We can't prove that there's one thing that causes the other. An easy way to identify correlations is observational studies. Basically, observational studies are just showing us that we know that group B, an example of figure 1, group B had a decrease in these genes. Do we know why? No, but we do know that there's some sort of mathematical relationship. Having a higher rate of cancer leads to decreased PER2 and BMAL1 expression. Causational studies, however, this is when we know that this thing directly is causing this other thing to happen. On the MCAT, the easy way to identify them are these studies that are going to be using knockouts, or they could also be changing the variable concentrations. So for example, in this experiment, notice in this first experimental group that we only knocked out BMAL1. The only difference between this group and the control group over here is the lack of BMAL1. And once you get rid of BMAL1, cancer prevalence increases. So therefore, you directly know that BMAL1 has some sort of role in tumorigenesis, specifically as a tumor suppressor gene. Same exact thing with the knockout of PER2. So these knockout experiments can help us prove causation. They could also do the same thing with modulating how much BMAL1 is expressed. So maybe if you decrease how much BMAL1 is expressed by a half, maybe then we'd see a 50% increase in cancer. Basically, if you start to kind of graduate the concentrations of something and then the actual net effect is also graduated, has a slight increase or whatever that matches your change in concentration, that means that there's a causation as well. Those are the two easy ways to prove a causation. So looking at option 3 says there's a causation, and the second experiment is in fact proving a causation. So because of that, we can eliminate answer choices A and C because they do not have option 3 in them. So notice from here, I don't even care about what option 2 states because the only thing that's left is looking at option 1. I'm basically just assuming that option 2 is false. Obviously, you could go through and figure out whether it's true or false or not, but we already know that it's false, not only because we've already identified that PER2 and BMAL1 are tumor suppressors, but also because it's not even an option that we have left through process of elimination. Option 1 states that the first experiment demonstrates that cancer causes decreased expression of PER2 and BMAL1. So looking at this, notice that when we have cancer, there's decreased expression of PER2 and BMAL1. Sounds good, right? So we're going to pick answer choice B. No. Stop yourself. Notice again that answer choice 1 says cause. This is not a causation. This is an observational study. Observational studies do not help us with causation. I am begging of you to get this through your head that on the MCAT, they're going to throw this at you at least once or twice, either in this section or especially the psych and soc section. There's a huge difference between correlation and causation. You have to make sure you are checking on that whenever you're looking at the results of a study. So we're actually going to get rid of 1, and our answer is D. All right, now that my cortisol is wearing off a little bit, I promise I'm not actually mad. Like, that's a really common mistake that people make. And we have been kind of hammering it in quite a bit in this one. But now that my fake anger is kind of gone, the last thing that I just want you to take home from this is that you do also need to understand this idea that if you remove a tumor suppressor, that's going to cause cancer. And then if you promote a proto-oncogene or mutate it into an oncogene, that's going to cause cancer as well. All right, moving on to question 4. Urea is a common denaturing agent that is produced mainly in the liver as a byproduct of protein breakdown and ammonium removal. Researchers note that upon adding a 0.002 molar concentration of urea to cells, northern blotting of ABK significantly decreases. Which of the following mechanisms explains the most likely cause of the decrease in ABK? Okay, so there's a shit ton of fluff in this one. The whole start of the question, I don't even care about. The first sentence, okay, this is what urea is. Great, whatever. It's a denaturing agent. The second one is basically just saying that if we add urea, northern blotting of ABK decreases. Northern blotting is measuring RNA, so basically we're just saying that urea is causing there to be less ABK mRNA. The ABK gene is being expressed less. And then basically we're just looking for why does this happen? Again, there's a lot of words in this answer choice, but it's all fluff. It's literally just saying, why is urea causing ABK expression to decrease? That's it. That's the root of the question. So more specifically, what in the passage is causing that? So this is a passage-based question. So just to kind of make sure that my brain is mentally prepared for a passage-based question. In the passage, the things that we talked about DNA, we talked about RNA, we talked about proteins, and we talked about hormones. Obviously, that's a lot of different things that it could be. But basically, the thing that I'm trying to highlight is that it's not going to be something like a glucose increase. Passage never talks about glucose. Passage never talks about fatty acids. Passage never talks about being shocked by a lightning strike or something like that. Basically, make sure that you're picking an answer choice that still applies to what the passage is talking about. That's one of those important things that I always think through going through the process of elimination. But even still, the first thing that we're going to do is eliminate choices that are not scientifically correct. This is the easiest thing to do. The easiest way to be a hater is through your scientific knowledge. So because of that, we can immediately eliminate answer choice C, because C is stating that if we block PER2, that is what's causing ABK to decrease, which is actually the opposite of what we'd expect. BMAL1 leads to ABK expression. That's pretty explicitly stated in the second paragraph. And PER2 is going to completely inhibit BMAL1 from being expressed in the first place. So if you block PER2, that means there's nothing stopping BMAL1 from being expressed. So if urea were preventing PER2 from actually getting into the nucleus so it could do anything, BMAL1 is going to have a heyday. It's going to be able to do whatever it wants. And after it forms that heterodimer, we're going to have a whole bunch of ABK in the cell. So C is wrong because it just gets the whole negative feedback loop wrong. Then looking at D, urea disrupts the structure of BMAL1, causing it to no longer function in its goal of promoting mitosis. This is also one that we can state is scientifically incorrect because that is not the goal of BMAL1. BMAL1, its job as a gene is as a tumor suppressor, which is to stop cell cycle progression and check to make sure everything's okay. Then once clock binds to it, that basically inhibits the whole functioning of BMAL1, allowing cell cycle to continue. So D basically is just misidentifying what BMAL1 is supposed to be doing, so we can get rid of that as well. BMAL1 does not have the goal of promoting mitosis. And then from here, we just need to eliminate any choices that don't answer the question or apply to the passage. So now whenever we look at B, just for example, B is trying to say that having a low amount of this urea prevents it from associating with transcription factors necessary for promoting PER2 expression, which is great. Like, as far as my brain is working, I don't think there's anything scientifically wrong with that, but that doesn't talk about ABK at all. It's talking about PER2. Whereas answer choice A mentions that urea is breaking apart that BMAL1 clock dimer that we know directly promotes ABK. We have a much easier tie into the passage and the question in answer choice A than we do with B, so that's going to be our answer. Now on to question 5, I promise only 2 left. Question 5 states, based on the information in the passage, which of the following is an organelle most likely stimulated by PER2 and BMAL1 to help prevent tumorigenesis, prevent cancer? Basically, we're looking for somewhere that is connecting these two genes to cancer. So it's probably going to be in that 4th or 5th paragraph somewhere. So again, identify the root of the question. Where in the passage can I connect the genes to cancer and some sort of organelle? 4th paragraph probably, because that's where they start talking about tumorigenesis. Notice right here in purple, literally the first sentence, where they talk about cell cycle progression, DNA repair, and apoptosis, and how BMAL1 and PER2 are really important in those two things. So this is going to be one that's going to be pretty hard to eliminate anything on the basis of scientific information, because that sentence mentions cell cycle progression, DNA repair, and apoptosis. And you can pretty much make an argument for any organelle being involved in any of those things. I mean, our scientific basis is still going to help us throughout the rest of this, but at least not in the same, much more direct way that it has with the other questions. So then we're going to be eliminating answer choices that don't answer the question. So let's go through this. We are looking for the most likely organelle that is stimulated by BMAL1 and PER2. Which of the following organelles is most likely involved in either cell cycle progression, DNA repair, or apoptosis, right? Okay, so peroxisomes. Peroxisomes are kind of weird. They do a lot of random stuff, but they're pretty important in metabolism. So that's not really a great answer choice. And obviously, metabolism is going to be quite a bit different in cancer than it is going to be normally, but that still just doesn't seem like a very direct connection. So I don't feel great about A. So let's just see if there's a better answer choice. Lysosomes are organelles that they basically just go around cleaning shit up. So that might be an okay answer choice. But again, we're trying to look for something that's directly involved in tumorigenesis, not necessarily tumor cleaning. So again, not really sure how I feel about that. Not great, but not horrible. Answer choice C, nuclear plasma membrane. Remember they talked about PER2 being translocated into the nucleus. So of course that is going to be our answer. Don't even need to look at anything else. We're just going to pick it and move on. Please tell me that your head just like pulled off sirens and like wanted to throw your TV onto a highway or something like that. Please never, ever, ever move on to the next question without reading all of your answer choices. I know it's a timed exam, but again, your job is to pick the best answer choice, the thing that is most likely stimulated by PER2 and BMAL1. Sure, you could argue that the nuclear plasma membrane is going to be important because they did mention translocation. That means it's a better answer choice than A and B because we kind of have a direct tie-in with the passage. That at least makes way more sense than what peroxisomes and lysosomes would be doing for cancer. But in that fourth paragraph that directly connected these two genes to cancer, they mentioned apoptosis. The mitochondria is the organelle that is largely responsible for apoptosis. Cells that have malfunctioning mitochondria or lack it at all really struggle to undergo apoptosis. So here they are telling us that BMAL1 and PER2, even though they don't give us an explicit way that they affect apoptosis, they are telling us that they have some sort of hand in apoptosis, which is conducted by the mitochondria. D is going to be the best option of the four. So again, I can hear you complaining from wherever you are right now. I'm in Texas. You're probably in Canada, if I'm being honest. Most of the people that watch my videos are in Canada for some reason. But basically, what I'm trying to get at, if I could just shut up, is that C is a fine answer choice. It's fine. There's not really anything wrong with it. It does make sense with the passage. It does have a tie-in with the passage. It does answer the question. It makes sense. There's nothing scientifically incorrect about it. It's just that D is a better answer choice. It's literally in the part that we just highlighted that connects these two genes to the actual production of cancer. You could maybe argue that the nuclear plasma membrane is involved in the cell cycle progression, DNA repair, and apoptosis, because I'm sure, like everything, it has some degree of effect in those three things. But the mitochondria is literally directly responsible for apoptosis in most of our cells. That fourth paragraph is telling us that BMAL1 and PER2 probably affect the mitochondria. Sure, there's plenty of other things that they affect, but the most direct response that we have is the mitochondria. That's why we're picking D. So again, that's why it's important to note that term or phrase, most likely. There could be multiple correct answer choice, but you're not just looking for a correct answer choice, you're looking for the best one. Even though C is like 95% of the way there. We do know that PER2 does have some sort of responsibility with the nuclear plasma membrane because it has a translocation. They specifically outlined how BMAL and PER2 affect apoptosis, not necessarily how they actually affect it, but that they do in some way. And the mitochondria is directly responsible for apoptosis. That's your answer. Again, you're looking for the best answer choice, not a correct one. Right, C is tempting, but it's just not as good. It's really close. But again, pick the best answer choice, not just any correct one. So D is our answer. And then we get to move on to our last question. So question 6, the last one, stating that given that BMAL1 weighs 76 kilodaltons and clock weighs 95 kilodaltons, which of the following correctly describes the results of native page using the BMAL1 clock complex, right? So they mentioned earlier multiple times that it's a heterodimer, dimer meaning two, hetero meaning that the two subunits are different from each other, which obviously they are. We have BMAL1 and clock. So they're just asking us what happens if we run this through native page. So again, that's the root of the question. What happens to a heterodimer in native page? So we're going to eliminate any answer choices that aren't scientifically incorrect. In the context of this question, we're basically just using our knowledge of native page to pick out what doesn't make sense. So like, for example, answer choice D says that proteins are too large to run using page. That's blatantly false. Page works fine for proteins. In fact, it's preferred for proteins. There's another gel called agarose. That is what we use for DNA and just like general DNA gel electrophoresis. So we can eliminate D. And then just to define native page is basically when we're not really adding anything to the proteins. There's no denaturing agent like SDS, or there's no reducing agents that are being added. So all of the protein interactions that are holding these two proteins together are going to stay. And because of that, it's going to still run as one big heavy dimer, BMAL1 plus clock. So because of that, all we have to do is just add the two weights together, and C is going to be our answer. If this were reducing SDS page, then the answer would be A, but it's not. It's native page, so the answer is C. So this is one of the easier questions, but pretty much it's just going to emphasize that you need to know your content. I've been pretty strongly emphasizing how important it is that you have a really strong content background, and obviously, this question is just a pretty good exemplification of why. So the final takeaways is that I want you to stop picking answer choices that have no scientific basis. There's so many times that one of my students will be like, I don't think this is true in answer choice A, but I think it's a better answer choice because it's more specific, so I'm going to pick that. Like, please just stop picking answer choices that have no scientific basis. That's incorrect. I don't care if an answer choice applies to the passage if it's trying to state that glucose is a protein. That's blatantly false. It's never going to be true, and there's nothing you can do to argue that it's true. So please just stop picking answer choices that have no scientific basis. Again, that's why your scientific knowledge, your content knowledge is really important. My other bit of advice that we didn't really work on that I think is really important to take home is that you should be finding a strategy that works for you, not for someone else. Again, please pick the best answer choice, not just any correct answer choice. Read all 4 answer choices, give them a good look in without being a creep about it. Be a hater, try to prove all of them wrong. Whatever is the best answer choice that's left over, that's the one that you're going to pick. And then finally, again, highlighting content stuff. In your practice, you should be identifying what content gaps you have. You want to limit those as much as you can, just so that it gets a lot easier to eliminate a lot of answer choices like we did today. Your content knowledge is important because it's going to help you prove the right answer, but it's also going to help you disprove a lot of other answer choices as well. That's typically one of the things that kind of separates the really high scores with the really good scores, is the ability to look at every single answer choice and say scientifically why it's incorrect. A lot of people will be able to tell you why something's correct, but not why every other answer choice is incorrect. You're trying to mitigate how many content gaps you have. One final recommendation is that I want to recommend getting you out of this thinking of high yield versus low yield. I know that on Reddit and a lot of YouTube pages and comments and your friends and textbooks and stuff like that, they talk about, oh, this is low yield. Just because it's low yield doesn't mean it's not going to show up. If it's low yield, it's entirely possible that it shows up. So I usually think about the content on the MCAT as it can show up, meaning that's something I should study, or it won't show up, meaning I don't need to worry about it. Sure, maybe the stuff that's lower yield, you don't need to spend as much time on, and you don't need to go as deep as the stuff that is quote unquote higher yield. But you still should be studying everything that might show up on the MCAT, everything that's in that syllabus that the AAMC releases. Don't just skip over stuff because it's low yield. I have a lot of students complain about, oh, my exam was super low yield because I had 5 questions about this obscure physics topic. It's like, well, yeah, sure, that physics topic is low yield, but it showed up. So I don't really know. Like, it sucks. I'm really sorry that sucks for you. It really does. I'm not being sarcastic when I say that. That really does suck. But again, even though it's low yield, it has a yield, which means that it shows up. So yep, that's it. That's my advice. If you guys do like this, I'll kind of keep making different ones just to kind of highlight different tips that I have in the future. And I will see you guys in my next video.